The helicase enzyme is going to unzip the DNA code. On one side, we have the leading strand. This will be built by the polymerase enzyme consecutively. As it is being unzipped, the polymerase is building a complementary base strand. On the other side, we have the lagging strand. This has to be built discontinuously. First, the primase enzyme will lay down a primer. The polymerase will then know where to go to start building the opposite complementary strand. Another primer will be laid down so the polymerase will continue to build up the strand. These sections that are being built are called Okazaki fragments. They will continue to be built down the strand as it is going to be unzipped. The primer needs to be replaced so that a ligase can replace the primer with its actual nucleotide so that it can end up with a consecutive strand. The end result will be two identical DNA copies that are identical to the original and to each other. Once the helicase enzyme unzips the DNA, the two strands go to the RNA polymerase, where the polymerase is going to match up the complementary nucleotides to the non-coding strand. When the mRNA is completed, it will be identical to the coding strands. The only difference is that mRNA uses uracil and not thymine. This happens within the nucleus, and the sequence has a lot of useless pieces that are called introns. These introns will be taken out of the sequence and stay inside the nucleus. All of the useful pieces called exons will be allowed to leave the nucleus. When the mRNA leaves the nucleus, they will go to a ribosome or RNA. The mRNA has sequences of 3 nucleotides that are going to match up with a tRNA's anticodon. The tRNA arrives at the ribosome and is placed where it connects the anticodon to match the codon. When this has happened, the amino acid will be released. This process is going to continue on to where the amino acids chain up. This tRNA exists and is going to be able to be recycled, so as this process continues, the tRNA will be able to grab another amino acid and come back to bring another amino acid in. This entire process will continue to happen until a stop codon appears, saying that the sequence is now finished. This finished protein sequence is called a polypeptide.
Oh, somebody messed up the, sorry, I'm talking loud.
Point out the polypeptide. Or repeat your last sentence. Point out the polypeptide. She has heard.
This finished protein sequence is called a polypeptide. This will code for whatever the gene expression is. This whole process is called the central dogma, which is going from DNA, RNA to proteins.